Welcome to this explainer. Okay, today we're tackling a topic that honestly strikes absolute fear into the hearts of so many AS level students. Yep, Edexcel AS Biology Unit 3. But don't worry, our goal today is to completely demystify these core practicals. Instead of just staring at some dry, boring checklist of lab equipment, we're going to build an intuitive framework to uncover exactly what the examiners actually want from you. So, get ready to totally shift your perspective. Because, well, here's the massive plot twist of Unit 3. How exactly do you test practical laboratory skills when you don't actually have a lab? It sounds like a trick question, right? But this is the literal reality of your Unit 3 exam. You're going to be sitting at a standard desk in a quiet exam hall with a pen. You are not standing at a bench with a beaker. And honestly, that completely changes how you need to approach your revision.
So here is our roadmap for today. First, the practical exam twist. Second, your core lab toolkit. Third, 6 must-know biology experiments. Fourth, cracking the Unit 3 exam. And finally, number 5, mastering the why.
All right, let's jump straight into Section 1, the practical exam twist, assessing practical skills in Biology I.
There is so much unnecessary anxiety around this unit. But the expectations, they're actually really specific. You are not assessed by doing practicals directly. Instead, you're tested through a written exam that's based on practical scenarios. A super common stumbling block for us is spending hours, literally hours, trying to memorize physical actions, like exactly how to swirl a flask or chop a potato perfectly, instead of just preparing for written, scenario-based questions. The examiner really only cares if you can explain the theory and the reasoning behind those physical actions on paper.
Okay, moving right along to Section 2, your core lab toolkit, covering the 4 pillars of the scientific method.
To actually survive these written scenarios, you need a mental framework, a toolkit, to fall back on whenever you're faced with an unfamiliar experiment on the page. Step 1, planning. Step 2, data collection. Step 3, data processing. And Step 4, evaluation. What's really cool here is that these aren't just random, arbitrary exam categories. This is the natural flow of literally any true scientific investigation in the real world. You plan your method, you grab your gear to collect the data, you crunch the numbers to process it, and finally you evaluate how the whole thing went. Every single question in Unit 3 maps back to one of these 4 steps.
Let's zoom in on Step 1 for a second, planning. To write a clear method or to do a proper risk assessment, you have to master experimental variables. You've got the independent variable, that's the one you are purposely changing. The dependent variable, that's what you actually measure as a result. And the controlled variables, the stuff you absolutely must keep exactly the same. Identifying these accurately is a huge stumbling block for a lot of people. If a scenario asks you to design an experiment and you don't explicitly state what you are keeping constant to ensure a fair test, you are dropping super easy marks.
Now, let's jump to the other end of the toolkit. Step 4, evaluation. This means identifying errors and limitations, suggesting actual improvements, and assessing reliability. Interestingly enough, this is exactly where the top scores are decided. I mean, it's relatively easy to collect data, right? But it is much, much harder to admit that your method or your data might actually be flawed. Spotting limitations in a given methodology and suggesting tangible improvements, that is exactly where you secure those high-level exam marks.
All right, on to Section 3, 6 must-know biology experiments. Basically, taking your toolkit for a test drive.
Now that we've got our 4 pillars established, we really need to apply them. Instead of just looking at a boring list of chores, let's treat these first 4 core practicals as strategic case studies for your toolkit. Notice how each one targets a highly specific skill. For enzyme activity, like tracking the effect of temperature on trypsin, the key skill is timing reactions and plotting rate graphs. With the beetroot experiment, which looks at membrane permeability, well, that's all about mastering the use of a colorimeter. Potato osmosis teaches you to calculate percentage mass change to track water movement. And microscopy? It isn't just about looking at pretty cells, it's practically applying the magnification formula and using scale bars. So you see, they aren't isolated experiments, they're targeted skill builders. Let's see how this builds when we compare the final 2 core practicals. On one hand, we have qualitative food tests, like Benedict's for reducing sugars, iodine for starch, or burette for protein. These are all about visual, color change interpretations. Is it blue or is it brick red? Versus, on the other hand, quantitative vitamin C investigations. Using DCPIP titrations requires exact, accurate numerical comparisons to track vitamin C content in juices.
The exam will absolutely test if you understand the crucial difference between making a qualitative observation and executing a precise quantitative measurement.
And because this is ultimately a written paper, you also need a deep theoretical understanding of your physical tools. Essential data collection apparatus includes microscopes for drawing biological diagrams and applying rules of scale, pipettes for ensuring accuracy in those titrations we literally just mentioned, and colorimeters for measuring things like pigment leakage in that beetroot experiment. You need to be intimately familiar with how to use these tools, and more importantly, why one tool might be way more reliable than another in a given experimental scenario.
Which brings us to Section 4, Cracking the Unit 3 exam, translating lab skills into exam marks.
So we have the toolkit, and we know the experiments. How does the exam board actually attack this on paper? Well, here are the precise written exam question types you're going to face. They'll ask you to design an experiment, identify the variables, interpret the data, evaluate the methods, and suggest improvements. Notice something familiar there? These map directly back one-to-one with our four-part toolkit we discussed earlier: planning, data collection, processing, and evaluation. When you sit down for that exam, just remember that every single question is pulling from one of those four buckets.
Finally, Section 5, Mastering the Why, the ultimate cheat code for Unit 3.
If you want to go from an average grade to a truly elite score, this is the fundamental mental shift you have to make. You must understand why each step is done, recognize common errors, and see how they apply to real exam-style questions. Just memorizing how to carry out an enzyme experiment, that's not going to cut it. If the exam twists the scenario and asks about a completely different enzyme you've literally never heard of, panic sets in for most students. But if you understand the underlying logic, like why you need a water bath to control temperature or why you need a buffer for pH, you can seamlessly adapt that logic to absolutely any tricky scenario the paper throws at you.
And this brilliantly brings us to our final thought. Don't just ask how, ask why. When you're revising your core practicals later tonight, I really challenge you to look at every single step in your methods and ask yourself, why am I doing this? What would happen if I didn't do it? Or what if I did it poorly? If you can confidently answer those questions, you aren't just memorizing a checklist anymore. You're thinking like a true scientist. And that is exactly what the examiner is looking for. So, are you ready to stop memorizing the how and start mastering the why?